Hello, everyone, and welcome to the SCCA's 2023 World Oceans Day Festival and our Nemo Talk series, the second in our Nemo Talk series this year. The Nemo Talks are brought to you by the Sunshine Coast Conservation Association, as well as Rhizome Up Media and the Green Film series. The SCCA works to protect biodiversity on the Sunshine Coast in the territories of the Squamish, Shishal, Pliamin, Klahoose, and Homolko First Nations. We are very grateful to the First Nations of this region for stewarding the lands and waters we share since time immemorial. We take inspiration from the respect, reverence, and connection that Indigenous peoples have always had with nature. And it is in this spirit of connection that we bring you our World Oceans Day Festival. Every year on June 8th, people all around the world come together to celebrate World Oceans Day. The United Nations designated this day to inform the global public of the impact of human actions on our oceans, to develop a worldwide movement of citizens for the ocean, and to mobilize and unite the world's population to support the sustainable management of our oceans. The theme of this year's program is Planet Ocean, Tides Are Changing. As our planet, our waters, and our waters warm and our seas rise, we must join forces to tackle the challenges ahead. This year's Oceans Festival program includes in-person and online screenings of thought-provoking and inspiring feature films and short films, interactive citizen science activities, and of course, these amazing Nemo Talks, all of which are geared toward inspiring, enlisting, and connecting people to work together and to take action to tackle the challenges ahead. We are so delighted to host Will Duguid from UVic and the Pacific Salmon Foundation to talk about forage fish and the Chinook salmon connection and how fish diet studies can help us understand changes in the marine ecosystems. I'm going to invite Diane Sanford to introduce Will for us.
Hey, so yeah, I'm very pleased to be able to introduce Will, who I met through my forage fish connections. So Will Duguid is a postdoctoral researcher working under Francis Juanes. I'm not sure if I'm pronouncing that correctly, at the University of Victoria, and he's leading the Chinook Salmon winter ecology component of the Pacific Salmon Foundation and BC Conservation Foundation led Bottlenecks to Survival study. He also initiated and continues to oversee the UVic adult salmon diet program. You can correct me on that if I'm a little bit off, Will. Will is a lifelong salmon enthusiast, having worked as a recreational fishing guide and as a salmon stock assessment biologist with LGL Limited, primarily in support of BC First Nations stock assessment programs. He completed an MSc, Master's in Science, in crustacean reproduction and development of a PhD in juvenile salmon, Chinook salmon marine ecology under the umbrella of PSF's Salish Sea Marine Survival Program. Today, will provide an introduction to key prey of Chinook and Coho salmon and discuss what salmon diets are teaching us about the food web that supports our salmon and the species that rely on them. He will also present new insights on migration ecology of Pacific herring and introduce current and upcoming research. And with no further ado, I'd like to invite Will to share his screen and do our presentation. And just as a quick aside, say that we met about anchovy that were happening in great numbers over by Somes actually area a number of years ago. And so that was how we bumped across each other. And that was a good meeting. Welcome, Will.
Thank you very much, Diane. Can everyone see that? Great. Okay, well, thank you very much to the SCCA and to Diane for inviting me here today. I appreciate it. The work I'm going to share is work completed in the Juanes lab at UVic. And specifically, my co-authors here, Jessica Qualley, Katie Ennis, and Wesley Greentree, did a fair bit of the research I'm going to be presenting. And a lot of this work is funded. There's various funders I'll acknowledge later, but the bulk of this work is funded by the Pacific Salmon Foundation. So I'm going to talk about forage fish and Chinook salmon. The picture there is one that I took. That's a Chinook salmon, probably about 30 centimeters, taken almost at this time of year, a couple of years ago in June in the northern Strait of Georgia. And the disturbed water on the right is age 0 herring. You can see a couple of little flashes there. So those are about 50 millimeters long, and that is that year's crop of age 0 herring. So that's a Chinook that's survived its first winter in the ocean and is chowing down on the bounty of the New Year's herring. So what I want to cover today, if I can advance my slides. There we go. It's a quick introduction to, or for many of you, probably review of some of the key forage fish species in the Salish Sea and in southern BC. And then I'm going to talk about the adult salmon diet program. So this is an initiative that we have had going in the lab since 2017 to work with recreational anglers to collect diet samples to understand the food web that supports salmon in British Columbia. And then from that, and from some of the results of that program, I want to segue a little bit into the story of non-migratory Pacific herring in the Salish Sea, their potential role in the ecosystem, and some exciting research that we're just starting to investigate this migration life history of herring here. And then some questions and discussion. And I think I'll probably be a little bit over 30 to 40 minutes with this, so might cut the question time a little bit short, but I'll do my best here. So coastal ecosystems that have a very dominant forage fish component are quite often referred to as wasp waste ecosystems. So this is a term that was coined by Bakun in 1996. And what it refers to is the fact that there is this narrowing at mid-levels of the ecosystem. And when I say narrowing, I mean narrowing to relatively low taxonomic diversity. So at the bottom of the ecosystem, one has a great diversity of different forms of zooplankton, including crustacean larvae and euphausids and copepods, and the larvae of other marine invertebrates, pteropods, all sorts of creatures. And then at the top of the food chain, there's a great diversity of predators, predatory fish, predatory birds, marine mammals, and often in the middle of the ecosystem, connecting these diverse lower trophic levels, these diverse upper trophic levels, there can be quite a low diversity of forage fish species. And I've shown 3 here, and there are 3 that I'm going to talk about: anchovy, sandlance, and Pacific herring, which are very important in the Salish Sea. And these species at the wasp waste of the ecosystem play an absolutely critical role in linking those nutrients produced at lower trophic levels to the higher trophic levels that require them. And because each of those species has unique biology, unique life history, unique reproductive timing and phenology, changes in the abundance, the relative abundance, or the loss or addition of its species at that narrow waist of the ecosystem have enormous implications for how the entire ecosystem functions. So this is the wasp waste concept, and I think it's a nice way of thinking about just how critical the role of forage fish is in the ecosystem. So what is a forage fish? Forage fish are characterized by very, very high fecundity. That means they have enormous numbers of eggs. And, in the Strait of Georgia, we're familiar, this is a shot from a satellite of the herring spawn occurring off Deep Bay and on the east side of Denman Island. These are spawning events that you can see from space. Huge numbers of eggs produced. They also have very, very, very small larvae. So the larvae of these species are not like a salmon fry. This is not a big individual investment in each egg. These are very, very tiny larvae, and the vast majority of them die. There's extremely high mortality. So there's an expression, the average larval fish is dead. And what this means is that populations of forage fish can fluctuate dramatically. And they can fluctuate dramatically based on environmental conditions and can go quite rapidly from very low abundance to very high abundance, and also from very high abundance to very low abundance. And they're very dependent on environmental conditions. And again, going back to the wasp waste concept, given their importance in linking lower and higher trophic levels, these shifts in abundance can have huge implications. So it's very important to understand what's influencing forage fish abundance and what forage fish are present in the system. So a few key BC forage fish species, and there's others here that I omitted in the interest of time, but I want to talk about three. The first of these is Pacific sandlance. Pacific sandlance are a really important species in the Salish Sea, same genus and very similar to sand eel in the North Atlantic. They are beach spawners. So they spawn in intertidal beaches in the substrate, and there's a network, as I know probably most of the audience here is aware, of citizen scientists who monitor those beach spawning locations, look for evidence of spawning in the form of eggs in the sediment, and finding those eggs demonstrates the linkages between these intertidal habitats and the pelagic food webs that they support, and provides a really important impetus to protect those habitats. So Pacific sandlance also have no swim bladder, which is interesting. And what this means is they have to constantly swim to maintain their position in the water column, which is a bit different from Pacific herring and other forage fish, which can equilibrate their buoyancy using a swim bladder. As a result of this, when there's not enough food available in the water, or when they can't forage, for example, at night, there's not a lot of point in Pacific sandlance continuing to hold their position and use energy swimming in the water column. So they actually bury themselves in the substrate. So this means they need to be close to habitats that allow them to bury in the substrate, that have suitable current velocities, depth, and sediment size for burying. So this limits their distribution to some degree. And also it means that they're unavailable to predators in the winter, because they actually bury themselves in the substrate during the winter during periods of low food availability. So this is an example of the sort of species-specific or group-specific ecology that has big implications for the food web. And Pacific sandlance are particularly important for seabirds in the Salish Sea and elsewhere on the Pacific coast. And there's quite a bit of work that's been done to characterize areas of high quality sandlance habitat. The figure on the left is from some work done by Cliff Robinson from DFO, which basically mapped suitable substrate on the bottom, suitable current speed, and suitable depth to identify hotspots that were suitable for Pacific sandlance to bury on the bottom, and therefore likely areas to find Pacific sandlance in the water column. And the figure on the right is from work recently published by Jacqueline Hurd, which actually used coastline data, substrate size, exposure, etc., to develop a spawning habitat suitability map for sandlance. So to identify beaches and intertidal habitat that was suitable for sandlance spawning, and then to validate that using the observations collected by all of these citizen science groups who are out there doing this work, collecting substrate samples and looking for sandlance eggs. So a really neat kind of example of citizen science at work to demonstrate the importance of these intertidal habitats. And I just was going to put a plug in that for folks who might be interested in participating, if anyone here doesn't know, which they probably all do, but anyone who's interested in participating in beach spawner monitoring, you can contact the BC Forage Fish Monitoring Network and be put in touch with your local group. And Virginia's email there is a good one, but there's other folks who can put you in as well. So northern anchovy, yeah, Diane, of course. Northern anchovy is another important forage fish species in the Salish Sea. And as Diane mentioned, this is an area of overlap that we had. Diane was very helpful in providing us with information on where anchovy were being observed and observations of them stranding in the middle of winter. And this was at a time when we were attempting to collate all the available information about northern anchovy in the Salish Sea. And what we found is it appears that if you go back through the records of northern anchovy observations and northern anchovy catch for the whole of the 20th century and the start of the 21st, we find a pattern where periods of elevated anchovy abundance in the Salish Sea appear to correspond to periods of warmer temperatures. So we are at the extreme northern end of the range of distribution of northern anchovy, and it is quite likely that as the climate continues to warm, we will see a continued increase in the consistent presence and abundance of northern anchovy within the Salish Sea. And at times, there are these major winter stranding events. So one of them was I think it was, 2019 at White Rock Pier. There was an enormous event where huge biomass of northern anchovy was at the pier for several days, was stranding on the beach, and was being fed on by all manner of birds and marine mammals. So quite a conspicuous and dramatic event when present. And so if you're interested in seeing the features to identify anchovy, you can Google Bob Turner's really excellent video on anchovy and how sound, which includes some really great underwater footage. So one of the interesting things about anchovy is if anchovy are increasing, what does it mean? Is it a good thing or a bad thing? And anchovies spawn throughout the summer. They have really small juveniles that are present throughout the summer, which could be a great thing. It could be an important food supplement to juvenile salmon in the Salish Sea, and I'll speak to that a tiny bit more in a second. But this is a really interesting and kind of obscure factoid, is that there's evidence for these widespread episodic thiamine deficiency events throughout the Northern Hemisphere and throughout not just marine food webs, but also terrestrial food webs. So thiamine, of course, is a B vitamin. It's an essential vitamin. It's involved in a number of different metabolic functions. And we don't know why. We don't know what's causing these thiamine deficiency events, but we do know that it's likely some form of anthropogenic forcing. We don't know what that is, but we do know that thiamine deficiency results either from inadequate dietary thiamine, and it can also result from eating food that's high in the enzyme thiaminase, which breaks down thiamine. And it turns out that anchovy are actually really, really high in this enzyme thiaminase. And it's possible, and this has been suggested for Chinook in California, that a diet totally dominated by anchovy can actually result in thiamine deficiency and potentially result in high mortality of juveniles. So really interesting, again, showing the kind of complex linkages that can occur with shifts in forage fish community composition. And there's some research coming up out of UBC that's going to be investigating this in the Strait of Georgia specifically. So then, of course, Pacific herring. Pacific herring are the dominant forage fish in the Salish Sea. The Strait of Georgia is the site of the largest spawning concentration, the largest spawning event for Pacific herring on the coast of British Columbia. And the Strait of Georgia population is the only remaining abundant and commercially harvested population out of 5 populations in BC. Herring are again an intertidal spawner. So in this case, not spawning in the sand, but rather attaching sticky eggs to vegetation in the intertidal and in the shallow subtidal. And the Strait of Georgia, and this will be important for what I'm going to talk about later, the Strait of Georgia herring population is primarily migratory. So for the most part, those herring, after spawning in late winter or early spring, they leave the Strait of Georgia and feed through the summer on feeding grounds on the continental shelf off the west coast of Vancouver Island. So they are considered to be a migratory stock. And they have potentially complex ecological roles as the dominant forage fish species in the Salish Sea. Typically, if you thought of what is the interaction between herring and salmon, you'd say, well, obviously herring are salmon food. And indeed they are. But herring are also salmon competitors. As juveniles, salmon eat the same food, the same zooplankton prey that herring do. Also, herring can at times be predators on salmon fry soon after they enter the ocean. Large adult herring do consume small fish. Herring can also be cannibals. Large adult herring can consume very small young of the year herring, and I've actually seen it occurring at really, really large scale off South Denman Island in the northern Strait of Georgia. And for juvenile salmon, the size ratio of herring to salmon can be really important. So in this picture, you see a juvenile Chinook that has fed on an age 0 herring, and you see a really typical situation where the front end of the herring is digested and the back end isn't yet digested. And this is a case where the herring has been eaten head first, but it's so large relative to the Chinook, very close to the threshold size that Chinook can consume, that it's only the front end that's really in the part of the stomach that's producing digestive enzymes, and the back end's hanging out into the esophagus. So this is something we typically see, and I've done quite a bit of work on this, seeing that it's only the largest juvenile Chinook that are large enough to consume that year's crop of age 0 herring. And so what this can mean is that where juvenile Chinook are too small to eat those herring, the herring may actually be a detriment, competing with them for the same zooplankton prey. But where either the herring are slightly smaller or the Chinook are slightly larger, suddenly they become an advantage as a really excellent source of fish prey. And in Puget Sound, this has actually been demonstrated, where we find that the growth of juvenile Chinook is far higher for bigger juvenile Chinook, but only where herring are present. So just to illustrate these very, very complex relationships between this dominant forage fish and their predators. So I think you'd agree that the forage fish species composition and abundance and how this is shifting through time is a really, really important thing to understand in order to understand the trajectory and health of the ecosystem. So this is kind of the impetus for this British Columbia Adult Salmon Diet Program, which started as a side project during my PhD work at UVic. We reached out to some anglers and said, hey, would you be interested in kicking in the stomachs of the fish you catch? And it's subsequently grown from there. So this is just the temperature anomalies from BC lighthouse stations. And this is just to come back to this point that we are in a period of change. We do anticipate that with a warming ocean and with other types of anthropogenic forcing, including ocean acidification and changes in precipitation and pressure systems, we are going to see changes in species composition. And in order to understand those changes, we have to be collecting data on what's going on out there. And the typical way we do this is with fishery independent surveys. And fishery independent surveys are important. They are powerful, and they have to be part of our toolbox to understand ecosystem change. So this is the Sir John Franklin, which is the new research ship on the Pacific coast. The challenge with fishery independent surveys is they're expensive. So going out there in these vessels and monitoring what's going on costs a lot. I think Franklin's probably about $65,000 a day to operate. So it means you can't be everywhere all the time, right? You do a survey once a year targeting a specific system, or you do a survey in one region over a longer period, but you can't be everywhere all the time. And that's where predator diet sampling offers an alternative approach. Predator diet sampling can be an inexpensive and spatially and temporally distributed way to understand changes in forage fish composition and ecosystem function. And a lot of this work is done with seabirds. So nesting surveys on seabirds, where researchers assess what prey those seabirds are bringing back to the nesting colony, is a technique that's been used for years and years and years. So our thought was, in the Strait of Georgia, we have BC, we have a near year-round recreational fishery. And we thought, hey, maybe we can engage some of these anglers as citizen scientists to contribute the digestive tracts of the fish they catch. And using these, we can understand changes in community composition and abundance. So that's the approach we've been taking. The objectives here were to address some basic knowledge gaps on Chinook and Coho salmon diets in BC. So what is the spatial and temporal variability in diets? Nothing has actually been published on Chinook and Coho salmon diets in British Columbia since the 1960s. You'd think this would be something we'd have lots of information on, but we actually don't. We wanted to use the salmon diets to improve our understanding of forage fish species ecology. We saw a value just in developing and maintaining a citizen science network, right? It's good to have fishery scientists talking to the folks who are out on the water and having that dialogue go in two directions as opposed to one direction. And then the really key objective here was develop a way to use salmon diets to monitor ecosystem change. So we do quite a bit of engagement and outreach. We write articles in fishing magazines to get people on board. We draw prizes. Every stomach that you contribute to the program is an entry and a draw for a reel, or Islander Reels donates a reel every year, and AP Tackle Works donates a spoon pack. We also send out hats or sweaters or shirts to people who contribute a certain number of stomachs. We provide an annual report every year to all of our participants on our results for the year. And then we also have copied the coded wire tag program from DFO, and we provide individualized angler reports. So every angler who submits stomachs gets a report back on the composition, the diet composition of the fish that they submitted and how that compared to the diet composition of all fish submitted from that same region and season. And when we've got this pipeline set up and we've got folks submitting samples, we get the samples back to the lab, we identify and measure stomach contents, we extract otoliths, so these are ear bones, which we can then use to reconstruct the size of prey that's too digested to measure. We also use the intestinal contents as a supplement to stomach contents to look at frequency of occurrence of different prey types. And we collect all kinds of physical samples for working with collaborators who are doing research on forage fish in other areas. So we started this in 2017. Pacific Salmon Foundation has been funding it since 2018. We have more than 250 participants, more than 4,400 stomachs analyzed, over 13,000 prey records, 4,600 otoliths archived from prey fish. And really importantly, we've had 13 mostly sort of early career scientists, recent graduates go through and get this science training through the program. And 4 of those have actually gone on to become fisheries graduate students in our lab group. And then we've worked providing samples to all sorts of different groups. We've provided herring for a project run by the University of Washington and NOAA to look at the population genetics of herring consumed by salmon. We've worked with DFO and Environment Climate Change Canada, looking at microplastic consumption in sandlands and forage fish contaminants. We've done provided some information for some research on the role of marine heat waves in changing sandlands size and abundance. We've provided data for end-to-end ecosystem modeling by researchers with NOAA. And then we're working with a bunch of groups to provide locally focused sample collection, where a First Nation or a community group is interested in diet composition within a specific region. So this just gives you a sense of the scope to date. The bubbles are the number of samples. So this is just Chinook. The bar charts show the number of samples by month at the top and the number of samples by year at the bottom, color-coded by region. So you can see we've got pretty good coverage of samples within the Salish Sea, some on the west coast of Vancouver Island, a handful from Haida Gwaii, but our best coverage is certainly within the Salish Sea. And as I said, over 4,400 samples to date. So a few results here. One of our questions is, what is the spatial structure of the Southern BC marine food web? And we had a graduate student in the lab, Wesley Greentree, who actually did some of this work as his honors. And I won't get into the details here, but basically what Wesley was interested in is, rather than arbitrarily grouping the Strait of Georgia or the Southern BC into different regions and asking, how does the diet differ between the regions? He actually created a grid, and he used a technique called cluster analysis to say, which of these grid cells group together? What does the data tell us about what regions are similar, rather than imposing regions and asking whether or not they differ? And so Wes did a really nice job of this. And what he basically found was 4 clusters of grid cells. And there were 2 clusters, the light blue cluster and the dark blue cluster, that were both really dominated by herring. And so one of these is the Strait of Juan de Fuca, and the other is the Strait of Georgia proper. There was one cluster of a single grid cell in Howe Sound, or the approaches to Howe Sound, that was dominated by northern anchovy, or I shouldn't say dominated, but the indicator species was northern anchovy. The diets there were characterized by a higher proportion of northern anchovy in the diet. And then there was another cluster in Harrow Strait, Southern Gulf Islands area, where the indicator species was Pacific sandlands. And this agreed, certainly the sandlands distribution agreed really well with the distribution work that Cliff Robinson and others had done, looking at suitable burying habitat. And the anchovy distribution agreed with work that we had done, looking at other available sources of information about where anchovy are found within the Salish Sea. So we have these spatial patterns. Are spatial patterns in diet composition and fullness consistent through time, was another question. And so for this, we've been basically reporting data every year through the State of the Pacific Ocean process, which is a DFO reporting process every year. And so you can find updated versions of this figure in those reports. But what this figure is showing is as you move from left to right, you have the regions indicated in the map at the top. So we have Juan de Fuca on the left, Gulf Islands, Howe Sound, Strait of Georgia, and West Coast Vancouver Island. The top series of panels are summer. The bottom series of panels are winter. And these are partial fullness indices. So what that means is rather than overall diet proportions, these show not only the composition of the diet, but also how full the fish were in total. So taller bars mean fuller fish, shorter bars are less full fish, and then the colors show you the composition. So what we see are some really consistent patterns. We see lower stomach fullness in the Strait of Juan de Fuca and Gulf Islands in Harrow than in other regions and seasons. We see consistent importance of squid. So these are invertebrates, which are mostly squid on the west coast of Vancouver Island in summer. That's the red. We see a really consistent occurrence of northern anchovy in Howe Sound in winter that doesn't really show up in other regions. Also, interestingly, a higher occurrence of surf perch, or a higher importance of surf perch, which is the light green in Howe Sound than in other regions. We see also sand lance being more important in Juan de Fuca. I should note that for this figure, to allow comparison between years, I've eliminated the months of May, June, and early July. And in doing that, have also eliminated the periods of highest sand lance abundance. So it's a bit misleading regarding sand lance abundance, this figure. If we include the whole year, we actually see higher sand lance abundance in this Gulf Islands Harrow Strait region. But what I really want to direct you to is the Strait of Georgia. So the Strait of Georgia has a consistently highest stomach fullness and consistently is overwhelmingly dominated by Pacific herring. So higher stomach fullness, consistently high stomach fullness across years, and overwhelmingly dominated by Pacific herring. So herring are very important. What is the size and age of herring that are being consumed? So here, this is where I mentioned the use of otoliths to reconstruct length. What we do is we use a digital software to measure otoliths automatically. These are the ear bones. And then we use the otoliths from herring that are intact and we can get a good length on to develop a regression relationship. And then for fish that are too digested to measure, we use that regression relationship to predict their length. And from this, we can basically put all of our herring into size categories and in turn into age categories. And when we do this, this is a very, very similar figure to the one I just showed you in terms of summer, winter, and all the regions. But now the bars are showing you the proportion of different age groups of Pacific herring. So age 0 is in green, age 1 is in yellow, and age 2+, so these are adult herring, are in this gray blue. So what you see is in the Strait of Georgia, in both winter and summer, diets are dominated by age 2+ Pacific herring. So these are effectively adult fish. And as you recall from when I was talking about Pacific herring earlier, this is considered to be primarily a migratory stock. In the summer, they are primarily off the west coast of Vancouver Island feeding. So the fact that diets are dominated by adults in summer is kind of interesting, right? These are, by their definition, by the fact that they are present in summer, these are non-migratory herring. And this, in the bottom right, is a figure showing the distribution of age 0 and 1 herring in diets. So the gray circles are the sample size for all fish examined in summer. The red shows you the proportion of those that contain age 0 and 1 herring. You can see age 0 and 1 herring are really found everywhere. They're quite distributed. If we look at the distribution of age 2+ herring, you see this really high importance in the northern Strait of Georgia in summer. So this is really cool. What we have is the Strait of Georgia being a region of high stomach fullness, and that high stomach fullness being driven by herring, and those herring being adult non-migratory herring. So really, really cool. So from this, I want to move on and I want to talk a little bit about herring. And first, I'll just acknowledge the funding we've received for the Adult Salmon Diet Program. Like I said, primarily the Pacific Salmon Foundation, but also World Wildlife Fund Canada, Fisheries and Oceans Canada, and Project Watershed. So out of this very interesting non-migratory herring results as part of the Adult Salmon Diet Program, we've built a research program, and Jessica Qualley is leading this research program. She's a master's student in the lab. And it's an investigation of non-migratory herring in the Salish Sea. So as I mentioned earlier, Strait of Georgia herring are managed as a migratory stock. The juveniles rear in the strait, but the adults are considered to migrate to feeding grounds on the continental shelf in summer, with adults returning to the strait in fall and overwintering prior to spawning in spring. But this is the figure I just showed you. There's a bit of a disjunct here because non-migratory adult herring are a really dominant critical ecosystem component in summer. But we understand very, very little about this non-migratory component of the population. There's anecdotal evidence of long-term change. So traditional ecological knowledge and the knowledge of fishers suggest that adult non-migratory herring were really, really abundant in the Strait of Georgia in summer historically. Anecdotally, in the early 2000s, they were almost entirely absent. And now recently, they are being seen. So this is just a echogram from some of the survey work we've been piloting. And we're seeing these really, really large, really deep water aggregations in summer of herring that we believe to be adult herring. So really pretty cool. But we have essentially no information, no data on changes in the abundance of this population component. So these are our problems. For one, we've got no way to distinguish migratory and non-migratory herring. If you find them on the spawning grounds, you have no idea if a herring migrated to the West Coast or not. We have no understanding of what factors may influence whether a herring becomes migratory or non-migratory, and we have no monitoring efforts to estimate that adult non-migratory biomass. So we had these research goals. One was to develop a natural tag for migratory and non-migratory herring so we could tell them apart. Another was to identify factors influencing the frequency of the life history types. And a third is to estimate summer biomass. So for the natural tag, what we've been looking at is isotope compositions, typically stable isotopes of oxygen and carbon. So as the otolith is formed, it's a mineral structure and it becomes a permanent record of the environment that the herring was experiencing at the time the otolith formed. So our hope is that the ratio of oxygen and carbon isotopes in the otolith could tell us where a fish spent most of its growing time, whether that was on the west coast of Vancouver Island or within the Strait of Georgia. And then we also wanted to identify whether growth, so how quickly these little guys are growing in their first year, could influence whether they migrate or not. And so for that, we can actually measure how big the otolith was before its first annulus. And you can see this dark line on the otolith is the first annulus, the first winter slowdown in growth. So by measuring this distance, you can say how much did this guy grow during his first year. So in order to do this, we've been doing a bunch of sampling in the strait, and Jess has been leading this. So we actually go out there and we use a sabiki rig with a large weight to get this down to, often as much as 100 meters, using sonar to locate schools. And we're collecting these herring. This gets really tiring, right? Reeling in 100 meters all the time. And Jess is actually, to save money, because researchers are always poor and we don't want to buy $1,000 electric reels, she's made these jigs so we can use cordless drills to retrieve our halibut reels, which gets you some really funny looks when you're in a group of people fishing. So we've been sampling these non-migratory herring in summer using jigging and then getting herring from other sources as well. So this map just shows Jess's efforts to date. The green dots are our jigging efforts to obtain herring in the summer. The orange is samples from DFO's roe herring test fishery. So we're getting samples of the spawning population, which may contain both residents and non-residents. And then the purple are samples collected either by jigging or from DFO's West Coast Vancouver Island integrated ecosystem trawl surveys. And these are providing an index for us of what a migratory herring looks like on the West Coast. And so some very preliminary results, and this is only for 10 fish from each group, but it's very, very promising. So the green ellipse, these are the oxygen 18 and carbon 13 ratio values of herring in the green ellipse from the west coast of Vancouver Island, in the purple ellipse that are resident in the Strait of Georgia, and then in the red ellipse from the spawning population. And it appears that the fish on the west coast of Vancouver Island have greater on the west coast of Vancouver Island have greater carbon-13 and oxygen-18 ratios. So this is suggesting we may actually be able to separate out migratory and non-migratory herring on the basis of their oxygen otolith values. Encouragingly, these values correspond to what one would expect. You expect that carbon-13 will be enriched in areas with nutrient-rich upwelling zones, so that would be the west coast of Vancouver Island. You expect that oxygen-18 will be inversely proportional to salinity and temperature. Sorry, let me get that right. So I'll show you the data and maybe I'll get my head straight on it. So this is Amphitrite Point on the west coast of Vancouver Island in purple and Entrance Island in green. These top two lines are salinity. So Entrance Island has consistently lower salinity over the time series due to the influence of the Fraser River. So the Strait of Georgia is less saline, and these dashed lines at the bottom are temperature. The Strait of Georgia is warmer. So it is warmer and less saline. And as a result of being warmer and less saline, it has lower oxygen-18. But what we're seeing in the herring otoliths is reflecting what we would expect from our basic understanding of how oxygen isotopes are sequestered in the otoliths of these herring. So this is really, really exciting. If we are able to look at a herring and say, is this a migrant or a non-migrant, when we have it in hand based on its otolith chemistry, we can then collect these from fisheries, or we can collect these from spawning sites. And we can say, in this fishery or at this spawning site, what are the relative contributions of residents and non-residents? A lot of First Nations, a lot of community groups are concerned that specific fisheries may be disproportionately influencing residents. So for example, the food and bait fishery in Stewart Channel or in the Discovery Islands, or roe herring fisheries in peripheral spawning areas outside of the core Denman Hornby spawning area. Are these fisheries disproportionately influencing the resident migration life history type? We can tell a resident from a migrant using their otolith chemistry. We can collect samples from these fisheries and we can address these questions and potentially manage fisheries to better conserve that very, very important resident migration life history. Also, we can then take the otoliths that we know to be residents or migrants, and we can look at first year growth. And we can look at this across years and we can say, does the rate that these juvenile herring grow in their first year determine whether or not they become migrant or resident? And this is a very, very small sample size, but just from the handful that we have in hand, it would suggest that fish that become migrants may actually have grown faster in their first year than fish that become non-migrants. But this will be involve a need for a much larger sample of spawners to confirm or refute this. And then finally, like I said, we have no data on the biomass out there in summer. And so we are planning to work with Strait of Georgia First Nations to develop a program based on small vessels to get out and survey some of known hotspots for non-migratory herring in summer. And using these sonar surveys, we're hoping to develop minimum biomass estimates. And these minimum biomass estimates can then be related to the spawning stock biomass estimates that DFO conducts during the roe herring fishery. Just so we can get a handle, is the number of herring we're seeing out there potentially 10% of the overall population? Is it 5%? Is it 50%? Right now, we have no idea. And hopefully we can also build a biomass index that through time we can use to assess change. So in conjunction with our adult salmon diet data, we can say in future, if that non-migratory herring population declines or disappears, we'll be able to detect that and we'll be able to understand what the consequences are for the ecosystem, which is not something that there was any data to do in the past. So we're planning to do this with active acoustics, so sonar from a small vessel, combined with jigging to confirm the presence of adult herring, and then also combined with drop cameras to take images of the schools and actually measure the size of the fish to confirm not only the species composition, but also the size of herring we're sampling. So to recap, we're attempting to develop a natural tag, or Jess is attempting to develop a natural tag to separate migratory and non-migratory herring. We're identifying the factors that can influence the frequency of these migration life history types. And then we're hoping to build this program with First Nations to estimate and monitor the summer non-migratory herring biomass in the strait. And it's really exciting. We have just with the BC Shrift announcements received notice of BC Shrift funding for this work. So we're really, really excited about this and really stoked how it's all been coming together. So Jess's work is funded primarily by the Pacific Salmon Foundation and MITEX, with lots and lots of support from Fisheries and Oceans Canada. So that's some of the folks who've been involved supporting Jess. And I should say also the Adult Salmon Diet Program. I mentioned the funders, but there are lots of folks who participated in the Adult Salmon Diet Program over the years. Lots of volunteers in the lab, lots of different processors in the lab, and also all of the fishers who collect these samples and collect the data on data cards and take the time to drop the samples off. Some of these folks are days of unpaid labor into this effort. So it's very, very much appreciated. So I think I will leave it there.
Will, that was amazing. Thank you. We're just going to get you to unshare your screen. My mind is so full of information right now. I can't believe all of the amazing work you guys are doing. A couple of things that struck me before we move into the Q&A is that you said that there wasn't really any research going on, and so no information since the 60s. And clearly you're filling a huge gap here with so many partnerships and so much good work. So I'm sure folks have lots of questions. I know I do. So we're going to move into the Q&A part of the program. I'll be taking questions in two ways. If people want to use the raise your hand function in Zoom, there's the little reactions button at the bottom of the screen. If you click that smiley face, you can push the button to raise your hand. You'll queue up and I'll call on you. Or if you don't want to ask in person, you can pop a question into the chat and I will direct the questions to Will. So we'll just give folks a chance to raise their hands if they want to. Or, okay, there we've got Ian. Go ahead, Ian.
Yeah, thanks. Well, that was amazing. I had no idea the depth of research being done throughout the Salish Sea and Strait of Georgia. It's incredible. You know, to think that I look out on it all the time and don't even realize how much stuff is going on there. It's amazing. One thing that popped into my mind was, given that there is the sort of international waters, you know, part of it's Washington, part of it's BC. Are there any challenges presented by the fact that you have different regulatory bodies and governing rules around how Canada and the US treat the environment and how that affects the monitoring in different areas as well?
Yeah, that's an interesting question. It's something that I'm not terribly familiar with, but I would say there are certainly efforts to bridge that gap in terms of communications among groups. And I think the Salish Sea Marine Survival Project, which was a PSF-led Pacific Salmon Foundation in Canada, and Long Live the Kings in the States, which is a organization in Washington. That program ran from about 2014 to 19 was the sort of official core of it, 5 years. And there is continued follow-up activities going on. That was an active attempt to bridge those gaps and bring together researchers across the border and to try to standardize research protocols, you know, to try to bring, for example, zooplankton sampling protocols in line so that data could be used across the border. And I think it certainly had some success. I think bringing research together was super valuable. I don't think it's led to 100% data standardization. That's a real challenge. In terms of regulatory frameworks, I'm less familiar. I mean, I certainly, for example, there's no active fisheries for Pacific herring anymore in Puget Sound. And so that could be argued to be a good thing, but it also, when that happens, it changes the priority of the management agencies, and less resources can be devoted to assessment, if that makes sense. And this is this is, a challenge for lots of important species on our side of the border as well, like sandlands, where, sandlands will get less stock assessment attention than herring because they're not a commercial species. So it's a bit of a rambling answer. I don't know if that got where you were going, but
Yeah, no, that helps. It's certainly, like I say, just in terms of even, protection of species and how that's treated, you'd hope that there would be some sort of concerted effort by both countries, but I can see, depending on who's in power, that can change on a whim, right? So yeah.
Yeah, and that's always one of the really big challenges, is that particularly for government management agencies, it's the challenge of those shifts in priority.
Yeah.
Yeah,
Wow.
well, thank you. Keep doing the awesome work.
Thanks, Ian.
We've got Alan's iPad. There you go. Just need to unmute yourself, Alan. There you go.
Actually, I'm Kevin. It's my dad's iPad. And I'm here as a sort of an amateur, I guess, citizen scientist. But my question for you is, Will, is your organization, your people, and your research, do you coordinate with other industries? I'm talking about like the forest industry, or maybe the shipping for, noise in the Salish Sea, that sort of thing. Do you coordinate your efforts with any other industry's research to get an overall picture of the health of the food chain and the forage fish in the region that you're talking about?
That's a good question. Myself, not really. Our lab group is certainly addressing those questions. Specifically questions like vessel sounds and vessel impacts. We are actively working on that. There's a doctoral student in the lab, Kelsey Murchie, who investigates impacts of vessel sound on Chinook salmon behavior. But I wouldn't describe it as a coordination. It's not working with any specific industry. It's collecting data in the environment without active collaboration. I'm trying to think of examples of what you're describing where there is active collaboration.
So I mentioned the forest industry as well, because my concern, I follow, what's happening in that industry fairly closely. And so the degradation of forests, erosion and sediment washing into the, down the waterways into the, Salish Sea or other areas, maybe my thought is that has an impact on the food chain as well. And, that's why I'm asking about whether you coordinate with any other industries such as that.
Yeah, no, that is an important point, and there has been work that I'm not very familiar with certainly on turbidity. There's some of the folks out of the spectral lab at UVic who are using satellite technology to get at some of those questions. But whether it is coordinated with industry, I don't believe it is, probably. But no, those are certainly good points, and I share your concerns about forest industry practices and not only direct impacts on hydrology and obviously the way hydrology influences Pacific salmon in freshwater, but also turbidity and nutrient exchange and things as well.
Okay, thanks for that.
Yeah, it's a follow-up too, that maybe we can look into it a little bit more and see if anybody's sort of coordinating on this. And Leanne Ennis has just posted a couple of things. We have a Salish Sea marine survival crew of citizen scientists here on the Sunshine Coast. They're still regularly collecting data and have the best record of consistent contributions to the data set. And then UVic Maya Sierra Costa Spectral Lab.
Yeah, that's the group that has done some satellite-based turbidity work, but also primarily works on phytoplankton, assessing phytoplankton bloom timing and extent using satellite technology.
Right on.
We've been chatting with her as well about potential to use the same technology to investigate herring spawn events that may be missed by other methods. So that image I showed of the herring spawn from satellite, it is possible in some cases to identify spawning events using satellites. And one of the challenges in the more remote areas like mainland inlets is capturing spawn events, because unless someone's there to see it, it can be missed.
Yeah. Okay, Evan and Sophia.
Hi, yeah, I thought that was a super interesting talk, and I'm wondering how I can get involved. I spent a lot of time on the water, and I'm sure I could provide some stomach samples. And I also have a sabiki rig and a deep water reel. So I haven't done a whole lot of fishing targeting herring, but I'm sure I could figure it out. And I have a good sounder too.
Awesome. Well, email me. Fire me your email address, and I'll send you the information on the adult salmon diet program. So the adult salmon diet program, you know, we have instructions, and we can either mail you data sheets or you can print data sheets. And then it's just as simple as taking the stomachs out, filling out the data sheet, and freezing the samples. And then we have a few drop-off locations. And if those drop-off locations don't work, then we can arrange to pick samples up. And then on the herring stuff, yeah, we're always scoping. We're always scoping where we can get more samples from. So I would say I'll put you in touch with Jess Qualley, who's leading that work. And certainly, if you see aggregations of what you believe to be adult herring, we're interested to find out about them for sure, and potentially obtain samples.
Okay, well, that sounds good to me. I'll send you an email.
Awesome. And did you get I think it's in the presentation there somewhere.
Yeah, we're going to follow up with you, Will, and ask you for links and a little bit more information and contact info, I think, to like put up on our events website afterwards, so that if there are other folks like Evan who, you know, are willing to do that kind of stuff, then they have quick and easy access.
Perfect. I put my email in the chat as well there. So
awesome. Perfect. Thank you. Diane writes, Sunshine Coast Friends of Forage Fish are the local group sampling for sandlands and surf smelt spawning sites on shorelines. Diane, did you want to jump in and say anything?
There we go. Yes. Yeah. So we've been sampling for a number of years for establishing sites and having them mapped as well with the State of Georgia data center. And so we have positive sites in quite a few areas, and it's on the map. And so that's what our goal has been, to get them there so that they're on record. Yeah, and you know, if there's any times that we can be of assistance, you know, with your work, Will, we certainly would. Although the chances of us actually seeing the fish themselves are random, right? That's random in itself. But we're also always looking for people that are interested in coming out and sampling with us. We try to hit the summer and the winter seasons for the different spawn times. And I'll be doing a demo on Thursday down at Trail Bay at the site down by Friendship Park at 3:00. So if you want to come and see how that sampling goes, we'll be doing a demo, and yeah, come and see.
Awesome. Yeah, we'll post the link to that event as part of the World Oceans Day festival. There's a whole page on it, so we can share that in the chat and then on the website with folks. We've got another question, Denise Lagasse.
Yeah, I would just like to know if there's any practical kind of beach etiquette that would be helpful for waterfront property owners. I don't have waterfront, but it's just that the shoreline is really important, and it must go beyond just like don't harvest, you know, seaweed during spawning season. Are there other things that contribute to the integrity of a shoreline?
there are, and it's a good question. There are, and I'm not the expert on it, so I won't speak to it, but there is a program through the Pacific Salmon Foundation, the Resilient Shores for Salmon, I believe it's called, that is providing education specifically on that subject to property owners and developers around BC. So I bet you somebody on this call probably knows more about it than me. But I will, I can look up the link and provide the link to that. But there certainly are educational resources out there on that subject.
Yeah, and actually last year, one of our Nemo talks was focused. We had the District of Seychelles planning staff who came and talked to us about basically development permit areas and the regulations around waterfront development and how to act on the shoreline. So that was development pieces of it. Not, they didn't really talk about, picking up seaweed and that kind of thing. But it was really about building on the waterfront and the rules and regulations around that to try and protect the foreshore.
Yeah.
So that we can, Ian, maybe you could pop a link to that if you could grab it off the website into the chat as well.
Another thing to take a look at is the Green Shores program through the Stewardship Center of BC.
Yes.
Yeah,
excellent.
these are all pieces. I mean, that establishing spawning beaches as well, all of those pieces, if they're, established and they're mapped, and they can perhaps be pieces that are added into the development permit areas, so that we can at least consider what is there and what's going on those beaches before we develop, or when we're developing, in the process.
So that would be my question, is like, if there's a good spawning beach, like maybe signage that indicates that, just so that it kind of comes into consciousness that, oh, there's things under our feet that matter.
Well, I think, Denise, my thought on that is that signage, no. I think it has to go back into the process when you're applying for permits. It has to be incorporated in that process, so that facet of the development that is incorporated, so that that's a spawning beach, and that has to be taken into account, so that the development incorporates that protection, or takes into account the timing for, to minimize the impact on the spawning, or it minimizes the impact on the actual area of the spawning. You know what I mean? So that municipalities or regional districts or local governments have to begin to be responsible for the areas that are sensitive, and that has not been taken into account up to this point in time.
Yeah, that's actually. Oh, go ahead.
So I was just going to say that I think advocating for good stormwater management is another worthwhile avenue, given that there is evidence, particularly herring larvae and eggs are very susceptible to hydrocarbon contamination. And so, whenever possible, being an advocate for stormwater management that prevents discharge of reverse flush or flush events off roadways and things into intertidal habitats is very important as well.
Yeah, this is such an interesting piece. We've actually got a campaign focused around local government environmental protection tools, and this is all part of the conversation of looking at a coast-wide sort of scheme of development permit areas that look at the interactions of all of these things, from the stormwater to the impacts on the beach and all the environmental values. So it's such a great conversation. One last thing, if you have time, Will, I know you're sort of pressed, but it was, I saw you had this big mapping piece, and I was curious about, in Howe Sound, we've got the Marine Reference Guide that's now the Marine Stewardship Initiative. It's got all kinds of, science and data and research that's going into it, but also some of the more like anecdotal pieces, traditional knowledge, that kind of stuff. So I wondered, is your project, do you guys have something similar? Are you engaging the fishers to add some extra stuff, not just guts? Yeah.
Yeah, we don't have, we haven't collected more. I mean, we do have some observations from fishers that come in with the data on the fish. We haven't actively incorporated those, but I didn't mention it, but the diet data can also be explored through Pacific Salmon Foundation's Marine Reference Guide.
Okay.
So That is, yeah, it's available. It's on the Strait of Georgia Data Center. And there's also, and yeah, I can provide you those links if you like. I should have mentioned there's also an ArcGIS dashboard to explore the data as well. So yeah.
Awesome, because those are just such crucial planning tools, honestly. So when the local governments are developing their protection measures, they can have access to up-to-date data. That's so crucial. I think we're going to call it. We're 7 minutes after, and I just want to say thank you so much, Will. That's just like, what an amazing body of work you guys are doing. And thank you so much for taking the time and coming to talk with us and share about your work and all the follow-up we're going to do after. Like Diane said, we've got a whole series of events and films and other resources on our Sunshine Coast World Oceans Day website that we hope folks will check out. Diane's doing the forage fish sampling demonstration on Thursday, which is actually Oceans Day at 3 to 5 p.m. I think is the time in Sechelt. And that day we're actually having another live event and a celebration where we'll be showing a film called Uncharted Waters. And this is a film about the Marine Stewardship Initiative that used to be called the Marine Reference Guide, and some of the folks that are working on some of the restoration projects in Howe Sound. So we're hoping folks will come out to that and hear from the speakers and the filmmakers, and come to our next Nemo talk, which is tomorrow. And we're going to be hearing from Susan Anthony from the Sea Change Marine Conservation Society about eelgrass and beyond, protecting resilient estuaries on the Sunshine Coast. So each of our talks really sort of builds on and segues into the other. So we just really appreciate everyone taking the time to participate on our events and hopefully get inspired to protect the oceans and get involved with all of this good work. Ian is posting all kinds of links and resources to the websites and the different organizations that we're collaborating with in the chat. And I just want to do a shout out to thank our sponsors, the District of Sechelt, Sunshine Coast Regional District, and the Sunshine Coast Credit Union for supporting the festival, and to our amazing working group, Diane and Angela, and Billy Carroll and Rhizome Up Media and the Green Film Series for all your amazing work to bring this to everybody. So look forward to seeing you all. And thanks again, Will.
Thanks very much for the invitation. Appreciate it.
Okay.
Take care, everyone.
Bye, folks.